Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast. Ozcast. Simply head to ozcastnetwork.com for details.
Life is uncertain. It's okay to feel stressed, anxious, worried, or frustrated. It's normal. With CalHope's free and secure mental health resources, it's easy to get the help you and your loved ones need when you need it the most. Call our warm line at 833-317-4673 or live chat at calhope.org today.
Dr. Travis Brown, what is This Medical Life all about?
This is the pursuit of knowledge as we learn about diseases from the ancient times to the present day. These are the stories of medicine.
Dr. Travis Brown, we're talking Prion's disease today, also referred to as CJD or mad cow disease.
Yeah.
Do you need to move away? Because I lived in London during the time of mad cow disease, hence I'm not allowed to give blood. Is it contagious?
It isn't. We're sitting across from you, but yes, I wouldn't take your blood. So although it's hard to know what that is, but we will find that out a little bit later. But yes, it's we're looking at prion disease, and so this is our understanding. You'll see the evolution of knowledge about the disease all the way from the start, all the way through to mad cow. Right. And so, look, prion disease, one of those things, Creutzfeldt-Jakob disease. It's very strange because it's a household name these days, and yet it's an incredibly rare disease. So when we look at the name of prion, it actually refers to, it's sort of an abbreviation, and it's called a proteinaceous infectious particle. So the person who first discovered it called it prions. And look, in fact, it wasn't until the 1980s that we actually realized what it was, the discovery. So it's very late. And the people that, encountered it first from a Western society perspective they were Australian anthropologists in the 1950s. And this is because they came and to study a group called the Four People, and they were Eastern Highlands of Papua New Guinea. And their names were Ronald and Catherine Burnt, and they wrote about it. And this disease, the Four People called Kuru. Now, Kuru in the Four language is to tremble or to be afraid or to shiver. And so anthropologists studying everything they do about, a culture and diseases, they began to write about this disease called Kuru. And they found that, the Four People knew about it, and they had actually classified it into almost three different categories. And, one is the patient is able to walk, and the next is they're able to sit, and then the third is they're even unable to sit. And so it's sort of, they go into a bit of detail about it. It wasn't until the mid-1950s that some of the medical investigators come along to say, well, what is this disease? And in that, they added a prodromal stage. So before they started to have symptoms of, walking issues, they actually noted that the patients were getting sick before they had walking issues. And they would have things like headache and pain in their limbs and abdomen. They got weight loss. But strangely enough, they didn't have any fever, any sore throat. There was no cough. There was no rash. There was nothing they're getting symptoms. So they couldn't actually link, this is like an infectious disease that would then cause them to have this degeneration.
And these people had been having this for a long time?
Well, it's hard to know because they wouldn't have a written history. No. And so, the anthropologists actually put it down to the clash of culture. This was their sort of, coming into contact with Western culture, and therefore this was a manifestation of that. Mm-hmm. Now, clearly the medical people said, well, that's rubbish. Having that classical doctor, anthropologist discussion. But so again, they had this prodromal phase. And then you have, the ambulant phase. So, is able to walk. Now, the patients, when they started to get this, the 4 people, they could move around, but they walked awkwardly and they had an unsteady gait. They had difficulty standing, and then they would lose control of their full body movements. And so this was sort of an unusual movement problem that they were going. They would get tremors that would start from the trunk. This was exacerbated by the cold. And then they noticed some clawing of their toes and curling of the feet, but this was to try and maintain balance as they had difficulty walking. And then they would get to the point where they had to walk with a stick, and then eventually they couldn't walk with a stick anymore. And so that would lead you to the sedentary stage, is able to sit. Now, they had postural instability, body control issues. They would have a tremor and joint issues. And that would lead them then eventually to the terminal stage. The interesting thing about all of this is it happened quite quickly. And so this was quite a rapid progression. And so once they got to the unable to sit stage, the terminal stage, they were bedridden, they would have difficulty swallowing, they would be incontinent and eventually lead to death. And so, anthropologists were looking at this and they investigated, well, what did they think it was? Now, the 4 people being in the culture, thought it was sorcery. They thought that a sorcerer had somehow taken a part of the victim, like, hair or, even, nail clippings, or even at the point of, partially, eaten foods, and that would be wrapped in leaves, put into a swamp, and then shaken each day. And every time it shook, the person would get a little bit worse and worse until eventually they developed a tremor. And so the way you manage that is one of your relatives tries to find the sorcerer, either bribes them, intimidates them, or even kills them to try and release the power from your relative.
Is there a Medicare code for that?
Well, and this, it sounds strange to us, and yet the same thing is, it's just because we're hearing it from we would have thought the same thing 1,000 years ago,
Sure.
And so the whole point is, this was clearly not the cause. And so we come to the point where everyone surrounding, and this was quite obvious from even the literature at the time, there was a practice that the 4 people did, and a few surrounding tribes in Papua New Guinea did, and it was ritualistic endocannibalism. So one of the important aspects of the 4 people, and it was evident from anyone who was an observer of their culture, is that one of their rituals was distinctly different. And it was the 4 people and some of the surrounding tribes around them, was that there was cannibalism. And that this was a distinction between 2. There is exocannibalism, and that is the one probably most people are familiar with from a historical point of view, is that you would, kill and eat your enemy to absorb their power. This has happened throughout history.
This is in the cartoons where we see the missionary in the big boiling pot.
Well, yeah, it's that kind of, but it's that kind of, that process of, you're absorbing the enemy's power. And this one, though, was endocannibalism. Now, this was part of funeral rites, a grieving process. So, to get past, and, part of a funeral rite was eating a relative. And so when we're looking at this, these people getting this unique disease, well, everyone's going, well, clearly it has something to do with that practice. And look, it's important because when you're looking about it, when we get into this, it just has to be known, I won't get into the details of it. It's also eating some brain matter. So this is quite an involved process, the ritual that they were going through. And so, when people were looking at this, they looked at the pedigree. How are these people? Is it, an infective process? And they looked at the family tree. Now, people were linked, but not biological link. They were kinship associated, but not necessarily blood related. So again, it was no surprise. People started going, well, clearly it has something to do with this. They didn't know what,
Yes.
but that practice was certainly, flagged.
So not passed down by bloodline, but by habit and culture and practices.
And kinship,
Yeah.
but not necessarily, blood. And so there was a doctor here at this time. His name was Dr. Gajusek. It was Dr. Cameron Gajusek. Now, he was studying this, and he thought there was some sort of genetic component, but he couldn't get evidence to support this. Just as we said, there was something linked between families, but it wasn't genetic. And the breakthrough moment for all of this was when they published some of this information of this disease. Now, as we know now, it is a brain disease. And what happened was you start getting holes through your brain. And what these holes are is filled with this protein. Now, you'll hear a term called spongiform encephalopathy.
Mm-hmm.
And this is to do with the brain looks like a sponge, and it's to do with, encephalopathy. So it's a disease of the brain, a disease of the brain that looks like a sponge.
Yep.
And so that's under the microscope. And they actually reported these findings, and they took a microphotograph of this. Now, the hard thing about all of this is everyone's trying to find a cause. If it's passed from one person eating, then surely there has to be some bacteria or fungi, virus, rickettsia. And they even got to the point where they tried to grow it through embryonic hen's eggs, which is a common way to proliferate viruses. No one could find anything. And so when they published it, and they published a photograph, it was noticed in 1959 by William Hadlow, who was an American veterinary pathologist.
Ah.
And he read and saw it and said, this looks like Scrapie. And Scrapie is a disease of sheep, and this was first described in German literature in 1759.
Some sheep also suffer from Scrapie, which can be identified by the fact that affected animals lie down, bite at their feet and legs, rub their backs against posts, fail to thrive, stop feeding, and finally become lame. They drag themselves along, gradually becoming emaciated and die. Scrapie is incurable. The best solution, therefore, is for a shepherd who notices that one of his animals is suffering from Scrapie to dispose of it quickly and slaughter it away from the manorial lands for consumption by the servants of the nobleman. A shepherd must isolate such an animal from healthy stock immediately because it is infectious and can cause serious harm to the flock.
Now, what did William Hadlow, just to reiterate, he saw an image of the Kuru patient brain matter, and he saw and he knew what Scrapie looked like, and he said they look very similar.
This is just a random, accidental...
This was reading the journal at the right time by the right person. And so what happened in the 18th and 19th century is they had an epidemic of Scrapie. And vets investigated, and again, they couldn't find at this time any causative agent. Experiments of trying to get sick sheep and then trying to infect healthy sheep didn't work. And it was just like the healthy sheep didn't get sick. There was a problem. Their experiments weren't long enough. They would say you didn't get sick within a few months. Problem is this disease takes years.
Right.
And so their whole premise was unfortunately unfounded. So they did get sick, but they would have gotten sick a lot longer than what their experiment gave them. And so what were the vets able to find about Scrapie? Well, this disease was throughout the body. Whatever it was collected in the brain and spinal cord, and it caused these holes in the tissue in the brain. Now, there was a genetic component to this. Some sheep were susceptible, others seemed resistant. So the amazing thing about this was whatever it was really hard to inactivate, meaning that you could treat it with like formalin, you could treat it with any commercial disinfectant, even radiation, and nothing inactivated it. So what they found was that sheep would pass it on to their offspring, and it was either through their colostrum and milk, It was either through birthing fluids or the afterbirth, and it caused these holes in the brain when they examined it under the microscope. So again, that's why it's called the spongiform encephalopathy. And so Dr. William Hadlow, he published this perspective, and he also sent a letter to Dr. Gajusek, and he said, you need to do an experiment on this. It will have to be into a primate because that's as similar as it can be for a human.
Mm-hmm.
And so in 1963, Dr. Gajusek, he procured a specimen from a 4-boy who had died from Kuru. And he teamed up with a virologist, Dr. Clarence Joseph Gibbs, and they established a lab to investigate what they believed was a slow, latent, and temperate virus that caused Kuru. They got 3 chimpanzees named Daisy and George, and one was unnamed. George turned out to be Georgette, so it was sort of, But the experiment, what they did was they used the tissue from this 4-boy and they injected it into the brains of the chimpanzees. Now, it took 2 years before symptoms appeared in the chimpanzees. And the problem with this was that any time with animal experimentation, it's always just a difficult area. But what had happened was the scientists and the chimpanzees bonded. And so you've got these unfortunate circumstances, and we have journal entries here about a neurodegenerative disease in experimental chimpanzees, but the scientists have bonded with.
June 28, 1965. Does not appear to be as active as usual. Stays by herself in the corner. July 14, 1965. Appears to have the shakes. Trembles at frequent intervals. No sense of balance. Fell off stoop in cage. Moving around very slowly. Jaw hangs open constantly. July 15, 1965. Tremors continue. Fell off top of cage today.
And so that was Georgette. Daisy was experiencing similar symptoms, and the scientists were becoming increasing distress. The chimpanzees were being cared for now because of the symptoms around the clock.
September 20, 1965. I don't know how much longer this animal, Georgette, can continue to degenerate. She's starting to appear emaciated and grossly weaker. We fed her again by hand. September 27, 1965. I fed the animal by hand today. Managed to get 3 apples and 3 pieces of whole wheat bread soaked in milk and cream into her. I'm quite worried about fluid intake. October 7, 1965. Georgette remains apathetic, though appetite is good. I'm firmly convinced that this animal will never recover. My life is getting to be hell for fear of sudden death or complicating secondary infection.
So what they have is the Kuru. And so they have a neurodegenerative disease. It took a long time for it to develop, but it was a sudden and rapid disease progression. And so you're seeing the scientists who are doing the experiment really starting to struggle with what they're seeing. And here's the challenge with it all, is this is what people of the 4 people would have seen, that, sudden onset of people that they love starting to die, and die rapidly, deteriorate. And so the chimpanzees were put down, and their autopsies did reveal that classic appearance in the brain of these holes forming.
Spongiform, yeah.
That's right. And so Kuru and Scrapie were similar diseases. They were neurodegenerational. They had a long latent time, and then they resulted in sudden and progressive symptoms and eventually death. And so this forms now what we call transmissible spongiform encephalopathy. These are diseases that form holes in neural tissue. And we know that Kuru, Scrapie, there's other ones like mink encephalopathy, wasting disease in deers, all are part of this. And that leads us to the 2 most well-known ones that we know about, and this is Creutzfeldt-Jakob disease, known as CJD, and then mad cow disease. So this is called actually bovine spongiform encephalopathy, or variant CJD, and that's coming up next.
All right. I also sense a Nobel Prize in the offing. Was my intuition right? Is there a Nobel Prize on this?
There is a Nobel Prize in this. But before we get to that, we still have to dig into these diseases. So the Nobel Prize comes when they actually figure out what causes this. But if we take it back to Creutzfeldt-Jakob disease, now again, this is a really rare disease, and it's amazing that people know the name. And the reason why I'm sure everyone is because in the 1980s and 1990s, CJD came up as the mad cow disease that pretty much caused,
Oh, it was everywhere, but we were told not to eat steak, at least not rare. But I couldn't help it. Right.
That's right.
I'm a lemming.
So, that's when the vegetarians got their ultimate revenge, I guess, wasn't it?
Well, that is true. Although I must say the science is lemmings don't actually just walk off cliffs, so I shouldn't perpetuate a myth.
But look, if we look at Creutzfeldt-Jakob disease, look, you've got two names in one disease. You have Jacob, part of it. This was actually a paper from 1923, and it was written by a Hamburg neurologist, Alphonse Maria Jacob.
Did you say a hamburger neurologist? Because that would be quite fitting.
Hamburg neurologist. And look, he had 5 patients, 2 men and 2 women, between the ages of 35 and 51.
That's up to 4. 2 men and 2 women. Did you say 5 patients?
I did, so I must have said, okay, we'll go. Maybe, should we start again? Our maths is off.
No, I think we should keep going.
Well, they had 2 men and 3 women.
Thank you.
Does that add up?
Yep.
All right, very good. So I would have failed my son's grade 3 test. So, look, and what he wrote about was a previously unrecognized neurodegenerational disease. These patients, again, they'll be similar theme. They had disturbance in motor function. They got to the point where they were talking about speech and emotional impairment. They had personality change and memory issues. And they eventually got to the point where they were unable to move or stand, and they got a dementia. And they died between a few weeks and a year of the onset of these symptoms. And that's a really significant decline. We have also a Breslau neurologist by the name of Bernhard Creutzfeldt. And about the same time, he wrote of a patient that had this peculiar focal illness of the CNS. And Jacob saw this, article, and he said, oh, this is identical to my case. And this is why I ended up being called Creutzfeldt-Jakob disease. It was really interesting. The twist in medicine is because later Creutzfeldt said, it doesn't resemble what, case I had.
Oh, wow.
And then Jacob's, interestingly enough, only 2 of the 5 would actually fit the CJD definition that we have these days. So, CJD, almost a misnomer in that instance, but, that's medicine. Yeah. But again, the reason this it's still a very rare disease, is because of the epidemic of a prion disease of mad cow. And this was when beef cattle in the UK, when they ended up getting sick, but they were part of the meat processing component for us, for humans. Mm-hmm. And so if they actually started tracing this back, and they were able to get to 2 cows in 1986 that were showing symptoms, but they think that probably happened from the 1970s. And so, how did it get into the cows? No one knows. Someone said, was this a spontaneous disease, or was this a crossover from Scrapie that had somehow gotten into the cattle and then gone from there? Again, similar symptoms. So they would ingest it, they had long prodromal stage, and then they got this progressive neurodegenerational symptoms. But the difference is we're in an industrial society. So the cattle who died were ground up, and then other cattle were given a meat and bone meal.
Oh, that's right.
And then other cattle would eat it. And it was this cycle. But because you're producing meat on such a large scale, it pretty much go on an industrial scale of infections. And so you put prions into the mix and it can go everywhere. And so that feedback cycle was amazing. In January of 1993, the vets in the cattle were, noticed there were about over 1,000 cases per day. And in 1995, there was reported 14,500 cases, 2000, about 1,500 cases. So they're starting to get onto it. 2005, there was, 225, 2010, 11, 2015, 2. So they knew what was happening and they actually got on top of it, but it still took a long time.
Yes.
And so the interesting thing about all of this, or the amazing thing, is people know it because at the time, government and health experts were saying, the hamburger you just ate or the meat that you just bought from the supermarket in the UK may cause you a progressive, irreversible, untreatable neurodegenerational disease. And you would remember that.
I do. Yes, very effective marketing.
Well, everyone remembers that, But the problem is they didn't just say you will possibly, you could get it, but you may not. So who's going to get symptoms? Who's not going to get symptoms? How much? How much meat do you have to have eaten? But you can't donate blood. And so, it probably was poorly managed.
Yes. Do you know if we can be tested to still be able to...
We'll have to ask our guest whether we can actually be tested that. But
We'll do that.
look, again, if you look at the span of this, disease or infectious period, 1980s to the 1990s, meat was affected in the United Kingdom. There was, shut down of industries where they couldn't. And that was, I remember my parents-in-law were dairy farmers, and I remember on their photo wall a cartoon of two cows talking, and one cow said to the other, are you scared of mad cow disease? And the other one said, no, I'm a duck. And, just it was just, something that no one really knew how the disease was, who was infected, who wasn't. And it wasn't until 1982 when we got Stanley D. Prusner, who identified these agents. And his theory going into this, he started studying them in 1972. And his theory was this had to be a virus. This, his research going through it was this needs to be a virus. But the problem is his own research indicated it wasn't a virus. In fact, he's the one that gave it the term the proteinaceous infectious particle, the prion. And this didn't have any DNA, no RNA. So the whole premise of the infectious model didn't work. And the problem with that is it caused a huge controversy. Someone was coming in from the medical community saying, we have a new infectious particle. And this caused massive controversy because people going, no, you're wrong. Even up to the point that controversy existed when he received the Nobel Prize and beyond, which was 15 years later after the initial, and this is because people had staked their livelihoods on finding the Scrapie virus. And there is, a final, a quote from him, in his Nobel Prize acknowledging this controversy.
People often ask me why I persisted in doing research on a subject that was so controversial. I frequently respond by telling them that only a few scientists are granted the great fortune to pursue topics that are so new and different that only a small number of people can grasp the meaning of such discoveries initially. Dr. James Leyden is a general neurologist who works in Adelaide. He runs his own practice called Leyden Neurology and is our guest today on This Medical Life. James, welcome.
G'day, thanks for having me.
Now, people won't hear this, but as we started, I had a little in my voice, and I mentioned that because I lived in London in the early 90s. And this is at a time, of course, of heightened awareness of mad cow disease. And I believe I'm not even allowed to give blood these days. So I'm prefacing that so you can be gentle with me and this topic. And is there any hope for me to ever be able to give blood?
No gentility needed. The odds that you have anything wrong with you from that are incredibly remote. But there's a long history of caution among the blood bank. And so whilst there's any chance, no matter how remote, they're going to be extra cautious. So potentially one day as technology changes, but you can understand after HIV and many other things that people have to be careful.
Yeah.
But it's no reflection on you whatsoever.
Okay, so I can nurse my phobia about needles and I can now have cover.
There we go.
Yeah.
Now, thanks, James. Look, we're talking about prion disease. We're talking about CJD and variant CJD. Look, can you tell us what are prions?
So to answer that question, we've really got to go back to the origins of life. It sounds dramatic, but it's true. DNA is an incredible piece of nanotechnology. DNA is a code that sits within cells using amino base pairs that carries all the information of what a cell should be and what should do, indeed, who we are. But DNA didn't appear out of just nowhere. It evolved itself. And so it had to have precursors. And for DNA to work, it needs proteins. It needs proteins as the machinery to interpret their DNA, to carry out the instructions, even to code it. And so before DNA, there were kinds of proteins that were perhaps precursors to life, self-replicating proteins. And prions are self-replicating proteins that very likely pre-existed DNA. And indeed, because we evolved from this, we carry those proteins around with us. They've evolved not to cause us harm, but you could imagine cancer is where cells run rampant, out of control, with the wrong DNA. If the wrong protein starts to run rampant and out of control, we call that a prion disease.
That's an incredible explanation for prions. So then if they're with us, how are they causing disease?
So as you know with cancer, a cell has instructions. It should grow to this point, but no more. It should do this, but not more. And if it loses those instructions and just suddenly goes nuts, then that's what a cancer is. Now, if a protein that doesn't need any DNA to tell it what to do starts replicating, then no one can tell it to stop. No one can control it. And so it just keeps on replicating and replicating. And with this illness called Creutzfeldt-Jakob disease, CJD, it's the worst of the worst. Once the protein starts switching on, it just keeps on reproducing itself until the brain is dissolved. It's the equivalent of putting your brain in a blender. Unrestricted self-replication dissolves anything of the right material. One of the big fears, people developing nanobots, little tiny robots that happen at a quantum level. And the idea is that if you set them to self-replication, but you can't turn them off, they could dissolve the planet. And in a way, CJD is an example of that. So a protein suddenly has all the material in the brain it needs, and it just keeps on replicating without any control. Now, we know about CJD because it's the worst of the worst, but understanding infectious or self-replicating proteins is incredibly tricky. And there's a lot of people who believe that illnesses like Alzheimer's, Parkinson's, and many other neurological diseases to which we don't yet know the etiology are in fact prion diseases, just not quite so rampant on the scale of CJD.
That scale of growth and replication makes me regret the bravado with which I still ate steak during my time in London. Very sobering.
You're pretty safe. The odds are incredibly low, and we'll talk about that, I guess, in a second.
Yes.
Yeah.
Actually, I need to pick up on it because this is not the discussion I was expecting with regards to, because you mentioned two diseases there, which I would not put into this category. This was Alzheimer's and Parkinson's. Now, is there a suggestion that this is a prion disease, or is there a theory about this?
Very much so. So we know that both Alzheimer's and Parkinson's and many of the neurodegenerative diseases spread through the brain like an infection. This has been known for a long time. It's called the Braque hypothesis, and in fact, that's the staging system that you as pathologists use to look at what level Alzheimer's is by where it infects. And so it seems to travel through the brain. Indeed, with Parkinson's, we know that the origin does very much look like it's coming from the gut wall and then migrating into the brain. And people are working on strategies such as cutting the nerve to the stomach to see if you can stop the flow through. So it's not been proven. We know that things like Alzheimer's have protein-related diseases. Things like the amyloid, as you know, you see lots of amyloid. It doesn't appear to be the cause. It appears to be a marker. All of this is controversial. But the question is, if there is an infectious process, then an infectious protein or a replicating protein, i.e. a prion, could well be the cause. And that's actually pretty exciting, because if you could eventually find that, which is very difficult to do, and then you could find an antibody to it, you might put an end to these illnesses. So a lot of there's a lot of research going on, but it's very tricky because it's very hard to diagnose small amounts of replicating proteins when you don't know what to look for in the first place. It's a bit like being blind and trying to find a bird.
So then if there is some promise there, but right at the moment, where are we at with sort of managing these diseases? Is there anything that treats them?
In terms of CJD, which I guess is what we think of traditionally as a prion disease, and leaving Alzheimer's and Parkinson's to one side, because that's an area of perhaps more controversy to suggest that this is what could be going on. But if you look at CJD, there is no treatment whatsoever. The proteins, once they're replicating, are just completely out of control. There's no way that we know of yet to stop them. The key infection control issue is to try to stop getting infected in the first place. Well, I guess we'll talk about the difference between how CJD happens and the variant CJD, but the main issue is not to eat brains, human brains or any others. And it's a big deal because Kuru in Papua New Guinea was caused by the religious traditions of people eating their relatives' brains to kind of absorb their knowledge. And Western culture has much stupider ideas than that. So, by not eating human brains, by not eating brains of other mammals, is very smart. And when we think about brains, you've also got to remember that the brain and the central nervous system includes the spinal cord. So, the vertebrae of cows and things will often have a bit of spinal cord and attached to them. So you've got to be very careful. Neurologists will never really want to eat brains after everything that we know about this. And the other big problem is that protein can't be sterilized. So you can't sterilize surgical equipment. Can't be done. So you can radiate them, you can do anything you want to them. You cannot stop or you cannot sterilize. So if there's a case of CJD in a neurosurgical case, they have to throw all the equipment out.
This is staggering.
It's incredible. So, well, let's focus. Can you tell us really then what the difference is between CJD and variant CJD?
Well, it's a bit of an artificial discriminator. So the standard CJD that I'll see in clinic is that we've got a protein that is very similar or is essentially a prion that is doing something else, and it's not set to self-replication. And then suddenly, just by pure chance, the randomness of the universe, it folds in a slightly different way and then starts replicating. That's called sporadic CJD. And they're probably different. It's probably not just one type of prion that does this, because there are different types of CJD. For example, there's one called Heidenhain variant, which it starts off by mixing up your visual field so that you're seeing everything appears in different places. And it's very characteristic. But there are other variations as well. Then we get to variant CJD, which is just the word we use to describe the kind of CJD that you get if you eat one of these self-replicating proteins from another animal's CNS. It was bovine spongiform encephalopathy back in your days, Steve, or BSE.
Thank you.
And there are just some slight differences to the presentation and the symptoms, but not clinically significant.
Right. So if we focus then just on CJD, look, how common is this disease?
So we don't know, really, because we only know what we're testing for or looking for. And we think that it's about a 1 in a million chance per person per year. But it's very difficult to diagnose, and many people die very quickly and the diagnosis might not have been thought of. And, if I look back at my career, I've seen far too many people for it to be really 1 in a million. Mm-hmm. So we know the incidence is higher than 1 in a million because we probably miss a lot.
Mm. Okay, then now you did mention some of the classifications, and from my reading, I saw the disease is kind of put into general categories, CJD of sporadic, genetic, acquired. Is this a useful distinction? Is there any clinical relevance for these categories?
Well, sometimes it can be helpful in diagnosis, but so the sporadic type is a random misfold of the protein that could happen at any moment to any of us sitting here. So, if you want to look at which is 1 in a million. So anyone who says they're going to win the lottery, just tell them that they've got an 80 times that chance of just suddenly dying from CJD when they buy a ticket. So put things in perspective. So that can happen to anyone. We have these proteins. These proteins are performing functions inside our brain and in the immune system, doing things we have yet to understand. And they're most prevalent in the really data-heavy parts of our kind of biology. Now, it can happen to anyone, but there are certain variations of the protein that are slightly more predisposed to folding in that way than other people. And that's been termed a genetic predisposition. And we've only probably discovered some of those, not all of those. And then there's acquired, which happens if you eat brain, have other people's brain infused into your body, or you get it from a surgeon's knife. And for example, when they were before they could synthesize growth hormones, they would use horse pituitaries. And they would have to get many, many different horses. And when they pulled all of that material, then the chances of having one of those proteins there was much higher, and all it takes is one. And so there was a huge amount of cases of CJD that came about from that with a delayed onset, sometimes 10, sometimes 20, sometimes 30 years. It's important to understand where the acquired ones are coming from and then change our behavior or change the practice that's caused it. But and there are some slight different aspects clinically to each of the types, but it's not something that we look it up depending on in the individual circumstances. It's not something that we'd instantly have to hand.
So then, with patients, what's their signs and symptoms that you'll typically see?
So the typical situation where we consider it is a rapidly deteriorating patient, someone who's got what appears to be a very rapidly progressing dementia. And that's when we think of CJD. Often it's associated with brief muscle jerks and incredible fear to small stimuli and a cascade of a bunch of different neurological symptoms. But it can be very hard to diagnose, and there can be many causes for people deteriorating rapidly. So it's one of those things that you often think of, but it may not be the case. That's the actual diagnosis. It can be very tricky to diagnose.
So then if we move our focus to variant CJD, how common is that?
So I'm not an epidemiologist in this area, but it all depends on what you're doing. So, if you are eating cows that had bovine spongiform encephalopathy, then it would be a very high chance. But to put it in perspective, every animal presumably has about the same chance we have of having CJD. And if you eat the animal's brain, then you're very likely to get it. But in actuality, it's incredibly rare. So worldwide, since it was first really discovered in 1995, I think 220 cases or so have been reported. So, there are 7 billion of us here on the planet. Yeah. And it must be happening all over the world, but there aren't many places with the sophistication to be able to diagnose and understand it. So it wouldn't be just limited to the UK. It's just that we haven't fully understood it properly.
Do we know the duration of the disease?
It varies quite a lot. The incubation period can be a few years. That is that the protein's just sitting there replicating at low enough levels on its path, on a trajectory to exponential growth, but it doesn't cross a threshold of causing symptoms. It could be a few years, could be 10 or more years. But once it clinically appears, it's incredibly rapid.
Right. So we've already indicated this, but the prognosis for these patients?
Certain doom, certain death.
Is there Is there almost a dose dependent? I mean, I guess that's a hard question, but like, if you've eaten more, does that indicate that you'll get it earlier, or we don't have any idea?
So, well, I mean, it's exponential growth. So theoretically, it shouldn't matter. If you have one protein, just one, it's enough to do it. But the more kind of brains you eat, the more chance you'll come across the protein. Right. But presumably the protein has to survive the denaturing process in your stomach, and it has to be absorbed, has to get into the nerve cells in the stomach. So, it's got a pathway. So, and then, along that pathway, there's every chance it could be aborted. So, you could have many theoretical sort of ideas as to what might make it more likely to be more likely to get infected or not. But no one really knows.
I just don't find the eating of brains all that common, at least in Western society. And I don't know, do you have any sense on the popularity of brain eating among cultures, James?
Well, as I said, the spinal cord's attached to, your lamb chops have a tiny bit of, you've got the spinal canal in there. That's central nervous system.
Right.
So if you chew on a lamb chop,
which I did last night,
you've just eaten some brain.
Well, there you go, Steve. So, sorry, you've rocked Steve's world right at the moment a little bit now, so that's messing with him. But can I ask then, is there tests that we can do for these diseases, prion diseases?
Yeah, so there's a bunch of tests, and they're particularly unhelpful. So, like all of neurology, really a lot of it comes down to clinical skill, because the tests are really, sorry, Trav, but the pathology. So there are tests. So there's a everyone talks about this test called a 1433, which is a kind of protein that can be found in the fluid around the spinal cord. Now the trouble is that it's a very non-specific marker, and it's essentially a marker that would show up in your fluid if your brain is being dissolved. So it doesn't necessarily mean it's CJD. It just shows that something's very badly wrong. And of course, since you're doing it in people where something's already badly wrong, it's often not very helpful. Often the test is positive, but there's another cause for the illness. For example, very recently you had someone with disseminated lung cancer throughout the brain and spinal cord. It was unknown at the time, but they were deteriorating very rapidly, and that test was positive. There's an EEG test where you can check the brain waves, and there's a very particular pattern to the brain waves that happens. These little spikes that happen every second, but that only happens once the condition is very advanced and the diagnosis is usually fairly obvious. But what tends to be most helpful is the clinical assessment, and the MRI scan is actually quite accurate. In very big studies, there's the fusion-weighted imaging shows quite a distinct pattern, and normal MRI scan is said under most circumstances to effectively rule the condition out. And that's probably not quite as well appreciated as it could be. And it's got pretty big implications, because the minute that you raise it as a possibility, I mean, for example, one of the neurosurgical suites in this hospital was recently shut down for a period of months because it was raised as a possibility. And so that whole suite, all the equipment, all of the stereotactic equipment, all had to be isolated and not used until this case was fully evaluated and determined whether it could have been CJD. So the diagnosis is a real problem, and by and large, it's clinical still with some adjunctive help from imaging. But it causes to pull our hair out frequently.
Yeah. So I was going to say then, with the pathology cap on, so the really only way you can actually diagnose this is through autopsy then, is it?
Yep, that's correct. And of course, people aren't lining up to do the autopsies. So we have, even if it's raised as a possibility, it causes so much, because it often has to go to forensics, and they have to use specialized equipment in areas that, it's a really challenging diagnosis, that one. So very challenging. But so with the test, so it's a clinical diagnosis. Given sort of the information we find in some of the theories coming up about even other diseases, do you anticipate prion disease will be something you'll deal with more in the future? Well, I think we already are. I mean, I think we just don't know it. As I said, I think Alzheimer's, Parkinson's, a bunch of other conditions called the multisystem atrophies and the progressive supranuclear Parkinson's, a bunch of the other dementias and the tauopathies, and what represents a very big portion of the poorly understood neurology is very likely to be a prion type phenomenon. It may not be nearly as rampant, but that might also make it much harder to detect. So I think there's a whole prion ecosystem out there. I think there are likely to be prion. This whole world of protein life existed before DNA. We don't really know how to detect it, and it's probably still around in some form or other. So when you say, will I be dealing with this more often? Probably the question is, will we understand more? Because there's just so much more. There's a whole world around us that we don't have the senses to see.
And just in closing, I think it's fair to say there's a bleakness about what we've discussed thus far. And thinking of the GP in their practice, any little tips, insights? What would you put inside their minds when contemplating the topics we've been talking about today, and prions in particular?
Well, I think there's absolutely no need to be worried. My wife and I have a bit of an argument. I get quite stressed when I get on a plane, but I'm usually stressed in getting through passport control and getting in the plane. And then once I'm in my chair, I'm quite relaxed. My wife, on the other hand, gets very tense when she gets into the seat. And she's like, well, what if we have a crash? It's like, well, there's nothing I can do about that. I was just more worried about missing the flight. I think at the end of the day, these things are incredibly remote. There's nothing we can yet do about them. There are plenty of things going on in life at the moment, so many things politically, so many things around us that we can change. This just at the moment isn't one of them.
So the advice to a GP is to recline back in that chair and order an in-flight drink. I think that's what you're saying.
Exactly.
Dr. James Layden, thanks for joining This Medical Life. This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts via Twitter. Dr. Travis Brown is at Dr. Travis Brown. That's DR for doctor. And Steve Davis is at Steve Davis. Editing and production is by Tim Whiffen. Design is by Tom Buzenjut. This has been a PathNotes Proprietary Limited production.
Life is uncertain. It's okay to feel stressed, anxious, worried, or frustrated. CalHope can help. Access CalHope's free and secure mental health resources. Call 833-317-4673 or live chat at calhope.org.
Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast.
Ozcast.
Simply head to ozcastnetwork.com for details.